Hello everyone, I am Anamol CK, first MSc Biology, St. Mary's College. Today I am discussing about principal assay and clinical significance of transaminases, lactate dehydrogenase, creatine kinase and phosphatase. Transaminases. Transaminases are aminotransferase enzymes that catalyze a transamination reaction between an amino acid and alpha keto acid. In transamination, the NH2 group on one molecule is exchanged with the keto group on the other molecule. The amino acid becomes the keto acid and keto acid becomes an amino acid. Transaminases are widely distributed throughout the body. It mainly two types, AST and ALT. AST means aspartate aminotransferase and ALT alanine aminotransferase. Both are excellent markers of liver damage caused by exposure to toxic substances. AST found in heart, liver, kidney, skeletal muscle, and ALT found in liver and kidney, less amount in heart and skeletal muscle. AST transfer of an amino group between aspartate and alpha keto acids to form oxaloacetate and glutamate. ALT transfer of an amino group between alanine and alpha keto acid to form glutamate and pyruvate. Clinical significance. ALT and AST are clinically significant aminotransferase. Both are excellent markers of liver disease. However, ALT is more liver specific than AST. AST levels are also significantly increased in skeletal muscular disorders and pulmonary embolism. Second one is creatine kinase, also known as creatine phosphokinase. It is an enzyme expressed by various tissues and cell types. The enzyme CK is a dimer of two subunits called M, found in skeletal muscle, and B, found in brain. The method used to separate and quantify CK isoenzyme include electrophoresis, ion exchange chromatography, radioimmunoassay, and immunoinhibition. Principle: creatine plus ATP gives phosphocreatine plus ADP. In this reaction, CK catalyzes the transfer of a phosphate group from creatine phosphate substrate to adenosine diphosphate. Clinical significance: diagnose heart attack, evaluate cause of chest pain, determine if or how badly a muscle is damaged, acute renal failure. Third one is lactate dehydrogenase. This is an enzyme found in all living cells. There are 5 isoenzymes of LD referred to as LD1 to LD5. A typical LD sometimes can be seen in the serum of patients with cancer and in tumor themselves. Principle: in this reaction, LDH catalyzes conversion of lactate to pyruvate and back as it converts NAD plus to NADH and back. Clinical significance: LDH is used as a general indicator for the severity of acute and chronic tissue damage, used to detect and monitor progressive conditions such as anemia, including hemolytic anemia, megaloblastic anemia, and severe infections. It plays an important role in cellular respiration, the process by which glucose from food is converted into usable energy for our cells. Last one is phosphatases. These are enzymes which catalyze splitting of phosphoric acid from non-phosphoric esters, mainly two types: alkaline phosphatase and acid phosphatase. Alkaline phosphatase present in most tissues, richest source being osteoblast in the bone, bile canaliculi in liver, small intestine epithelium, kidney, placenta, breast during lactation. ALP plays an important role in growth and development of bones and teeth. It is useful diagnostic test in bone and liver disorders. Clinical significance: moderate increase in ALP level is seen in hepatic disease such as infective hepatitis, alcoholic hepatitis. Very high level of ALP may be noticed in extrahepatic obstruction caused by gallstones or by pressure on bile duct by carcinoma of head of pancreas. Drastically high level of ALP are seen in bone disease where osteoblastic activity is enhanced such as Paget's disease, rickets, osteoblastoma of bone, and hyperparathyroidism. Acid phosphatase. It is an enzyme found throughout the body, but higher concentration is found in prostate gland, which is known as prostatic acid phosphatase. Acid phosphatase test is primarily used to diagnose prostatic cancer and to monitor the effect of treatment. It may also be used to detect disorders related to the blood cells, heart, liver, and other organs. Principle: in citric buffer, ACP hydrolyzed the substrate, that is alpha naphthyl phosphate, to produce alpha naphthyl and phosphate at acidic pH. Alpha naphthol react with diazo-2-chlorophytoluene forming an azo dye-colored compound which absorbs maximally at 405 nm and is directly proportional to the total acid phosphatase activity. The color intensity is measured by colorimeter at 405 nm. The intensity of colored complex is directly proportional to the total ACP activity in the serum. Clinical significance, elevation level and condition. First one is severe elevation, prostatic disorders such as hypertrophy and carcinoma. Second, moderate elevation, Paget disease, carcinoma of bones, etc. Mild elevation, multiple myeloma, leukemia, thrombocytopenia, and liver disease. Reference. Then thank you.